In this video, we will discuss about some of the in vivo results of the CAR T cells with FAS disruptions. As in our previous videos, we have understood the results that are presented in this paper that points out that FAS negatively regulates the CAR T cells. As the CAR T cells gets activated, this will trigger FAS signaling. It will then cause apoptosis of the CAR T cells. So if FAS has been signaling is disrupted, then it might prevent the apoptosis in the CAR T cells and thereby increase its anti-tumor efficiencies. So this hypothesis will be tested in vivo in the mice model, and the approach or the experimental overview is shown in this panel A, where they use three different kind of construct. First, that does not have transmembrane domain, second one with transmembrane domain, and third one with transmembrane domain with the FAS disruptions. And they check the samples or check the mice till like 15 to, they check the survival rate of the mice till like 12 to 18 days or 20 to 25, 12 to 18 weeks or 20 to 25 weeks, depending on the mice health. So for that they took the CAR T cells from two different donors and make this transduce them with these three different construct, and thereafter they check the health of the and in the before transducing the CAR T cells, they transduce the mice with this virus NALM6B-ALL, and 4 days after transducing of this virus, they inject these CAR T cells, and then they check the tumor growth and the survival rate. So in terms of the tumor growth, they can see that in the case of control where there is no transmembrane domain, we can see the tumors goes almost shows in almost all the mice after 3 weeks, and they seems to have low survival rate as the tumor is already reached to its peak at after 3 weeks. However, in the case where transmembrane domain is also there, they can see the tumor survival rate is high, and in some case the tumor is completely eliminated. It looks like almost completely eliminated. In the case where FAS signaling is down-regulated, we can see further improvement in the anti-tumor efficacies, as all the mice seems to be survive after 6 weeks, and the tumor rates are also low, even comparable to even when it is compared to the TGF beta plus transmembrane domain without FAS signaling disruptions. And in terms of survival rate, we can see that the mice that have this transmembrane domain, intracellular domains, and along with, so this is intracellular domains. I might have mistaken it as transmembrane domain. So the mice that are transduced with CAR T cells with intracellular domains, and along with this where far FAS signaling is disrupted, we can see the highest survival rate compared to the case where FAS signaling is not disrupted. And if there is no intracellular domain, we can see a very low survival rate. And these results are shown in the panel C for the where the mice is transduced with one donor, and for both the donors the survival rates are shown in this panel D, where we can see that for both donor 1 and donor 2, survival rates are higher when they are transduced with CAR T cells with intracellular domain and FAS signaling disrupted. So this in together indicates that even in the in vivo, the relationship between FAS and is like negatively, as it relationship between FAS and CAR T cells is inverse. FAS is inversely related to the CAR T cells in vivo also, as we can see that when the FAS signaling is disrupted, it will increase the CAR T cells survival rate or CAR T cells treated patients, and that ultimately leads to ultimately because of its increased anti-tumor efficacy. And one of the reasons could be because CAR T cells are able to survive for a longer period of time, because the apoptosis, endogenous apoptosis that is triggered in the CAR T cells through the FAS signaling pathways is been disrupted. So I hope you understood how they validate their hypothesis about the negative relationship between FAS and CAR T cells. If you need in the in vivo studies, if you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.